Hi everyone, so in this video we're going to be going through both 8B and 8C because I think they flow into each other really nicely. So in our last video we learned about vaccination. In this video we're going to be learning about how these diseases actually emerge, what that means and how we can prevent the spread of these diseases. So some of this used to be, really new and exciting, but I think after living through COVID, which a lot of you probably did, all of this stuff seems, quite logical and probably is already in your brains already just from living through that time. So first we're going to learn about how diseases emerge and re-emerge and what that means for populations. So remember COVID, I'm sure we all do. So pathogens and diseases can emerge and re-emerge all the time. So emerge means to, be new. So emergence means to be new, like something like COVID, and re-emerge means something that was originally a disease that threatened people, went away and has come back. So both of these types of disease can threaten outbreaks of disease throughout the world. So just to highlight, so a lot of diseases are non-infectious and aren't caused by pathogens, and we're not talking about them in this course, things like diabetes, things that can't be passed between people. We're talking about infectious and contagious diseases throughout this whole course. So they're the only ones that we're going to be focusing on during this video, which are ones that can be spread through people. So we have learned about many different types of pathogens. So we looked a bit at bacteria, viruses, parasites, worms, all those sorts of things in our very first video. I think the important thing to know about these pathogens, regardless of what they are these two things. And you might have heard these words before when you were hearing about COVID stuff. How contagious are they? So contagious means how easily are they transmitted between people. So how easy is it to catch it from someone else? And how virulent are they? So this is how severe the disease is when you actually get the pathogen. So contagious, how easily it spreads. Virulent, how severe it is when you get sick with it. So if a pathogen is highly contagious and highly virulent, this can be severe and devastating to populations. So we can have things that are highly contagious but not highly virulent, like things like the common cold, which spreads super easily, but they're not really that severe to most people. But then if you get this double whammy of high contagion and high virulence, that's when you get really quite severe diseases happening in populations. So we talked about this a little bit. So what are emerging and re-emerging? So emerging just means something that's completely new and new to a specific population or new in recent times. So COVID is a perfect example of that. Also things like bird flu and things that have been, very new over the last, 10 or 15 years, they're emerging. But then we also have things that are re-emerging. So we have things like Ebola, we have things like measles, which we did almost eradicate through vaccination some time ago, but because people are choosing not to get vaccinated, is starting to come back. So those are examples of re-emerging pathogens, and they're both equally as, damaging. I would say emerging is probably a bit more concerning because we have no knowledge of it. So that's why COVID was so scary, because no one knew how to deal with it. So what happens if you don't respond to these emerging and re-emerging diseases? Well, three things can happen. This one's probably haunts your dreams, but the other two you might not know about. So what can happen is there can obviously be an outbreak. So an outbreak is just when an unexpected large group of people become infected. So you might often hear about outbreaks in like childcare. There might be like a gastro outbreak in childcare. That just means that it's unexpected and a large amount of people got infected with something. So that's like your normal, not as severe. Then we get into the more severe categories. So you can have an epidemic, which is a sudden and widespread increase of the occurrence of a disease in a specific population. So this is like quite specific. It's only affecting sort of one demographic of people or place or time. An example of this is the Ebola outbreak in Africa. So this was an epidemic because even though it was really devastating in Africa, it didn't really affect other people in other places. So that's an epidemic. And then you've got your creme de la creme of disease, which is your pandemic, which is what COVID was. So that's when it affects pretty much everybody. It affects spread all regions, affects a great number of people. And that's when it becomes really, really difficult to control. And COVID is a perfect example of that. A really, good but sad example of pathogens being introduced into populations and the devastating effects of that is when Europeans settled into Australia. So obviously we know that we had an Indigenous population of Australians that lived here before the European settlers came. So when those European populations came to Australia, they brought their diseases with them. And even though those were, super common diseases in Europe, those were completely new emerging diseases in Australia, which the Indigenous people had never been exposed to before. So when they introduced these diseases, smallpox was a massive one, this caused major disease and death in Indigenous populations. So you can see in this graph here, so this red line is the amount of colonists that arrived. So obviously that's generally increasing. And then we have the amount of Indigenous population is in blue. So you can see how that's slowly going down as the European population is going up. So you can see that these Europeans, they brought these diseases over and it caused really devastating effects. And that's why disease spread and also medical interventions like vaccinations nowadays, obviously that was not a thing back then, are really important to prevent these emergence of diseases in places that didn't originally have them. So why was this so devastating for Indigenous populations? I'm hoping that you could probably answer that by knowing what you know about immunity. So obviously, because this was a new disease that was introduced, they did not have any memory cells, they did not have any natural active immunity. So there's obviously those sorts of things. So they had never been exposed, so therefore they were all undergoing a primary exposure, which we know takes a really long time and can actually cause quite severe disease. Apart from that, there's also very little knowledge of management and treatment in those populations. More people means that there was more risk of spreading, and also changes to diet as well. And many of these negative impacts still affect Indigenous peoples today. So that's why it's so important to understand how these diseases spread and how they can affect populations. And this has formed the basis of many of the medical interventions that we'll talk about a little bit later on. So now we're going to look at how we can identify these pathogens before they can spread and cause, severe disease, and how we can prevent the spread of these diseases as well. And a lot of this is probably going to be second nature to you after living through a pandemic. So the first thing that we need to do to be able to prevent the spread of these diseases is we first need to be able to identify a pathogen. So, obviously very important. If we don't know what's hurting us, how can we treat it? So because pathogens are becoming more virulent and more contagious over time, which is terrifying, we need to act really quickly to avoid outbreaks, epidemics, and pandemics. And I think COVID has been a big wake-up call for a lot of people, and we would prefer not to have things like that in the future. So the first thing we need to do is be able to identify the pathogen, because then after we've identified it and we know what it looks like, then we can start mounting treatments that are going to be specific towards it. So there's a lot of different ways we can identify a pathogen. Obviously, we can look at the physical characteristics of a pathogen, the structure of bacteria and viruses, which we'll go through in a second. There's also a bunch of different lab techniques that you can use. You can use serology, and you can use ELISA assays, and you can use like agar plates, and you can sample them, and there's a bunch of different things that you can do to look at the immunological aspects of a pathogen, also look at the phenotype of a pathogen, and also the molecular makeup as well. So you can look at the genetic material of a pathogen. There's lots of different ways that we can identify them, but the main way that we want to look at for this course, we're going to look at the physical characteristics of pathogens, and you will need to know how to differentiate between pathogens using their physical characteristics. We're looking at bacteria and viruses here because they're normally the two main types that you're going to, need to deal with. So that's what we're going to be looking at. So you'll see on the bacterial cell here, we've got, the general structure. They're unicellular. They usually have a flagellum, this like tail thing that comes out here. They have DNA. They have circular DNA inside. They've got cytoplasm, ribosomes. They've got a cell wall. They've got this capsule that encirculates them. So there's a couple of different things that you can take from that. Then we come over here to the virus. So a virus has its DNA or its RNA, its genetic material inside, and then that is encapsulated by a capsid and then an envelope as well, a nuclear envelope. They can also look like the one on the right here. They still have the genetic material inside, but they can also have this neck and tail structure as well. So being able to differentiate between those is important. So how do we control the transmission of these pathogens? This is probably something you're very familiar with by now, but pathogens cannot survive alone and they need to move between hosts, which is what we call transmission between people. So this transmission can either involve contact with an infectious person, or it can be indirect. So it can be by being in close proximity. They could sneeze and an air droplet could fall on you. There's a bunch of different ways, and these are the main ones that we're going to be looking at. So there's of course airborne transmission. So if someone coughs, a particle can be released into the air and then you can inhale that particle. Droplets is the same deal from a cough or a sneeze. There's also of course physical direct contact. So obviously if you're touching the bodily fluids of someone else and then touching your own mouth or eyes, you're very likely to get it. And then there's of course like fecal oral transmission for things like gastro and stuff like that. So how do we control the transmission of pathogens? And I'm not going to spend too much time on this because I think you know. So the correct method of control will depend on the pathogen, of course. So things like COVID, where it's respiratory droplets and air droplets that are spreading that disease around, it would be things like masks and preventing the respiratory spread, things like washing your hands, vaccination, of course, for a couple of diseases, quarantine and isolation, which we all know very well, so that you don't get into contact with people. But there's also things like treatments like antibiotics, antivirals, food hygiene, all of those sorts of things that I'm sure you're very well versed in by now. So I hope this helps. If you have any questions, bring them to class and I will see you there.